Hi, everyone. Every living thing on Earth has one problem in common. How do you stay alive in a world that is constantly trying to mix with you? Think about it for a second. Cells are tiny bags of chemicals surrounded by an environment completely different from what exists inside them. Inside a cell, conditions have to stay relatively stable. The right ions, the right nutrients, the right pH, and the right amount of water. But outside the cell, everything is constantly changing. Without some kind of boundary, life couldn't exist. Cells wouldn't lose or would lose important molecules. Toxic substances would rush in, and the chemistry required for life would collapse. That boundary is the plasma membrane. What makes the plasma membrane is important um, is that it is universal. Bacteria have plasma membranes. Archaea have plasma membranes. Plant cells, animal cells, fungi, protists, they all have plasma membranes. Even though these organisms look dramatically different, every cell depends on this structure. The plasma membrane is much more than just a barrier. It controls what enters and leaves. It allows cells to communicate. It helps cells capture energy, and it creates the internal conditions that make it possible. In many ways, the plasma membrane is what separates living systems from the outside world. In this lecture, we'll explore how plasma membranes are built, why phospholipids form bilayers, how membranes control transport, and why membrane structure is one of the most important features shared by all life. By the end of this module, you should be able to explain how the structures of the plasma membrane allow cells to regulate interactions with the environment and describe how phospholipids, membrane proteins, and membrane fluidity contribute to membrane function and cellular homeostasis. When we zoom in on the plasma membrane, we find that it is primarily built from molecules called phospholipids. Phospholipids are unusual because one part of the molecule interacts well with water while another part avoids water. The phosphate containing head is hydrophilic, meaning water loving. It can interact with the watery environments both inside and outside the cell. But the fatty acid tails are hydrophobic, meaning water fearing. They do not mix well with water, and this creates a really important physical barrier. If phospholipids are placed into water, they automatically organize themselves. The hydrophilic heads face outward toward the water, while the hydrophobic tails turn inward away from the water. As a result, um, sorry, As a result, phospholipids naturally form a bilayer, two layers arranged tail to tail. This bilayer creates stable boundary between the inside of the cell and the outside environment. It is flexible enough to move and grow, but stable enough to keep the cell intact. And this structure is one of the most important features shared by all cellular life. To really understand why membranes form, we need to understand two important chemical ideas. That is, this idea of hydrophilic and hydrophobic. Hydrophilic substances, as we said, interact well with water. They may carry charges or have polar regions that allow them to mix with water molecules easily. On the other hand, hydrophobic substances are different. They avoid water because they are nonpolar and cannot form favorable interactions with water molecules. And water itself is actually what drives membrane formation. When phospholipids are surrounded by water, the hydrophobic tails are forced away from the water, while the hydrophilic heads remain exposed. This causes the molecule to spontaneously organize into bilayers. In other words, cells do not have to build membranes one phospholipid at a time. The molecule self assembles because of their chemical properties. That self assembly is incredibly important because it means membranes can form naturally, repair small damage, and maintain flexible boundaries around the cell. Phospholipids form the basic structure of the membrane, but membranes are not made of lipids alone. Embedded within the bilayer are many different proteins, and those proteins do much of the membrane's work. Some membrane proteins transport materials across the membrane. This is important because many molecules cannot simply pass through the hydrophobic center of the bilayer on their own. Other membrane proteins detect signals. They allow cells to sense changes in their environment and respond to chemical messages from other cells. Some membrane proteins carry out chemical reactions. In many cells, important metabolic processes happen right at the membrane surface, and other proteins anchor structure in place. They help connect the membrane to the cell wall, the cytoskeleton, or structures outside the cell. So while the phospholipid bilayer creates the boundary, membrane proteins make the boundary functional. One of the most important properties, pardon me, one of the most important properties of the plasma membrane is selective permeability. Selective permeability means that some substances can cross the membrane easily while others cannot. Small nonpolar molecules like oxygen or carbon dioxide can often move directly through the phospholipid bilayer. But many substances cannot pass through the hydrophobic interior of the membrane on their own. Charged ions, sugars, and many larger molecules usually require transport proteins to help them move across the membrane. This selective control is critical because cells must carefully regulate their internal environment. They need to maintain proper concentrations of nutrients, ions, water, and waste products, even when external conditions change. By controlling what enters and leaves, the plasma membrane helps maintain homeostasis, relatively stable internal conditions that allow cells to survive and function properly. Although all cells use plasma membranes, not all membranes are identical. Bacteria and eukaryotic cells mainly use phospholipids with ester-linked fatty acids. These are the membrane structures most commonly shown in biology diagrams. Archaea are different. Their membranes use ether-linked lipids instead of ester-linked lipids. That chemical difference may seem small, but it has important consequences. Ether-linked membranes are often more chemically stable and more resistant to damage from heat, acidity, or other extreme conditions. Many archaea live in environments that would destroy most other cells, extremely hot environments, high salt concentrations, or very acidic conditions. Their unique membrane chemistry helps them to survive in these extreme environments. So while all membranes perform similar functions, the exact structure of the membrane can vary across different groups of life. The plasma membrane is far more than just a thin covering around the cell. It performs several critical functions that allow cells to survive. First, the membrane defines the cell boundary. It separates the inside of the cell from the outside environment, creating an organized internal space where life's chemistry can occur. Second, the membrane controls transport. It regulates what enters and leaves the cell, allowing nutrients in, removing waste, and helping maintain proper internal conditions. The membrane the membrane also enables communication. Specialized membrane proteins can detect chemical signals from the environment or from other cells, allowing cells to respond to changes around them. In many organisms, membranes also support energy generation. For example, bacteria use their plasma membrane for important energy producing processes, and eukaryotic cells use internal membranes in organelles like mitochondria and chloroplasts to generate ATP. And all of these functions contribute to one larger goal: maintaining homeostasis. The membrane helps keep internal conditions relatively stable even when the external environment changes. Without membranes, cells could not maintain the organization required for life. All right. One important thing to understand about plasma membranes is that they are not rigid structures. The membrane is flexible and dynamic. Phospholipids can move sideways within the bilayer, and many membrane proteins can move as well. Pardon me. This flexibility is called membrane fluidity. Membrane fluidity is strongly influenced by the types of fatty acids present in the membrane. Saturated fatty acids are straight and packed tightly together, which makes membranes more rigid. Unsaturated fatty acids contain bends caused by double bonds. These bends prevent tight packing and increase membrane fluidity. Temperature also affects membrane behavior. At lower temperatures, phospholipids move more slowly and membranes become more rigid. At higher temperatures, phospholipids move more rapidly, making membranes more fluid. Cells must carefully balance membrane fluidity. If the membrane becomes too rigid, transport and membrane protein functions can be disrupted. If it becomes too fluid, the membrane may become unstable or leaky. Many organisms adjust their membrane composition to help maintain proper fluidity under different environmental conditions. For example, cells in colder environments often increase unsaturated fatty acids to help keep membranes flexible. So, although membranes form stable boundaries, they are also dynamic structures that can adapt to the needs of the cell and the environment. In this lecture, we explored the plasma membrane and why it is one of the most important structures shared by all cells. Membranes create boundaries that separate the inside of the cell from the outside environment. They help maintain homeostasis, support communication, and organize the chemistry required for life. But membranes also create a challenge. Cells still need to bring in nutrients, water, and other important molecules, and they must remove waste and maintain proper internal conditions. So, if the membrane acts as a selective barrier, how do substances actually move across it? That question brings us to the next lecture, where we'll explore transport across membranes, including diffusion, osmosis, passive transport, and active transport. All right. Um, now, when you with that, thank you so much for attending this lecture, and we'll see you at the next one.